Growth hormone. The name sounds self-explanatory. The pituitary releases it, and the body grows. But think about what that actually means. A gland about the size of a pea releases a microscopic chemical signal into your bloodstream. And somehow, bones lengthen, cartilage grows, muscle develops, organs grow. An entire human body changes size. How? By the middle of the 20th century, scientists had finally isolated growth hormone. They knew where it came from. They knew it was connected to growth. But now they faced the question hidden inside its name. How does one hormone make an entire human body grow? Finding the answer would take decades, and lead scientists through one of the most surprising chapters in the history of endocrinology. This is the History of Growth Hormone, Part 3. How does growth hormone actually make us grow? The first problem appeared almost immediately. During the 1940s, researchers had learned to extract growth-promoting material from pituitary glands. Among the scientists leading that work were Cho-Hao Lee, Herbert Evans, and Miriam Simpson at the University of California, Berkeley. They worked with pituitary glands obtained from animals, particularly cattle. The preparations contained a substance capable of producing striking growth effects in laboratory animals. And that raised an obvious possibility. Doctors were already treating some human hormone deficiencies by replacing the missing hormone. Insulin was the perfect example. For decades, insulin obtained from cattle and pigs had been used successfully to treat people with diabetes. So why couldn't doctors do something similar with growth hormone? Some children with severe pituitary disorders grew far more slowly than expected. If their bodies were missing a growth-promoting hormone, perhaps physicians could simply replace it. Researchers tried animal growth hormone in humans. And this time, the logic failed. The preparations that could stimulate growth in animals produced little or no meaningful growth response in humans. That was unusual. Scientists began discovering that growth hormone had a property that made it very different from hormones such as insulin. Its biological activity was strongly species-specific. Growth hormone from one species did not necessarily work in another. The molecular structure mattered, and cattle or pig GH could not simply substitute for the hormone produced by the human pituitary. That left researchers with a difficult problem. If they wanted to find out whether replacing growth hormone could restore human growth, they needed human growth hormone. There was only one place scientists knew they could obtain it: a human pituitary gland. During the 1950s, researchers began developing methods to extract and purify growth hormone from human pituitary tissue collected after death. For the first time, human growth hormone was becoming available for clinical investigation. One of the scientists who changed that was Maurice Raben. Raben was an American physician and endocrinologist whose work focused on hormones produced by the pituitary gland. By the 1950s, researchers could obtain human pituitary glands during autopsies. The gland itself was tiny. The challenge was extracting and purifying enough human GH to actually use it. In 1956, Raben published a method for preparing growth hormone from human pituitary glands. 2 years later, he put it to a clinical test. In 1958, Raben reported treating a 17-year-old boy with severe growth failure associated with pituitary deficiency. It was human growth hormone, extracted from human pituitary tissue. The treatment continued for months, and the boy's growth rate increased. For physicians, this was a remarkable moment. A child whose own pituitary could not provide the normal hormonal signal for growth could be given that missing human hormone, and growth could resume. Other researchers followed. Children with growth hormone deficiency were treated with pituitary-derived human GH, and many showed substantial increases in growth velocity. Researchers could measure the changes in height. They could follow skeletal development. And they could observe changes associated with activity at the epiphyseal growth plates, the regions of cartilage near the ends of a child's long bones where lengthening occurs. After decades of investigating the pituitary, scientists finally had compelling human evidence that replacing GH could restore growth. But that success brought them directly back to the question we started with. They could now watch a child grow after receiving the hormone. They still had to explain how it happened. And to answer that, researchers would turn away from the whole child and begin studying a small piece of growing tissue, cartilage. By the late 1950s, researchers had crossed an important threshold. They could give human growth hormone to a child who was deficient and measure an increase in growth. But measuring the result was very different from explaining it. To understand why, scientists turned their attention to the places where children's bones actually become longer. The epiphyseal growth plates. A growth plate is a layer of specialized cartilage located near the ends of a developing long bone. Within it, cartilage cells called chondrocytes divide, increase in size, and help produce the cartilage framework that is eventually replaced by bone. This continuous process allows the bone to lengthen during childhood and adolescence. So researchers now had a more precise question. Growth hormone was released into the bloodstream. But what happened when that hormonal signal reached growing cartilage? Did GH travel to the growth plate, bind to the cartilage cells, and directly stimulate the processes required for growth? That seemed like a reasonable explanation. But it needed to be tested. At Washington University School of Medicine in St. Louis, physician-scientist William H. Daughaday was investigating the relationship between the pituitary gland and tissue growth. Working with him was a young researcher named William D. Salmon Jr. Together, they designed an experiment that reduced the enormous complexity of a growing body to something they could examine directly: a piece of cartilage. If growth hormone acted directly on growing cartilage, they should be able to see its effect there. Salmon and Daughaday needed a way to measure what growth hormone was doing inside cartilage. They chose cartilage from the ribs of young rats. But these weren't ordinary animals. Their pituitary glands had been surgically removed. Without the pituitary, the animals had lost the normal source of growth hormone and other pituitary hormones. That gave the researchers tissue in which the normal pituitary influence had been greatly reduced. Now they needed a measurable sign that the cartilage was responding. Cartilage contains large molecules called proteolycans, which are important components of its extracellular matrix. Their production involves the incorporation of sulfate. So Salmon and Daughaday supplied the cartilage with sulfate labeled with a radioactive isotope, sulfur-35. The radioactive label allowed them to measure how much sulfate the cartilage incorporated. In other words, they now had a biochemical signal they could actually count. Then came the critical test. They incubated the isolated cartilage with growth hormone. The prediction was straightforward. If GH directly stimulated the cartilage, adding GH to the tissue should increase its incorporation of radioactive sulfate. The experiment was designed to turn the question, does growth hormone directly make cartilage grow, into something measurable. They ran the experiment, and the result forced them to reconsider the entire model. The result was not what the simple model predicted. When Salmon and Daughaday added growth hormone directly to the isolated cartilage, it did not produce the increase in radioactive sulfate incorporation they were looking for. But they had another clue. When cartilage was exposed to serum from normal animals, sulfate incorporation increased. Something circulating in the blood was stimulating the cartilage. So they compared that with serum from animals whose pituitary glands had been removed. The activity was dramatically reduced. That made sense. Without a pituitary gland, the animals no longer had their normal supply of growth hormone. But then the researchers did something crucial. They gave growth hormone to the animals before collecting their serum. And when that serum was placed with the cartilage, the growth-promoting activity returned. Think about what that meant. GH added directly to the cartilage had failed to produce the expected response. But GH given to the living animal caused something to appear in its blood that could stimulate the cartilage. There had to be another step between growth hormone and the response they were measuring in growing tissue. Salmon and Daughaday proposed that GH was causing the body to produce another circulating substance. They didn't yet know exactly what it was, so they named it for what they could measure: sulfation factor. For the first time, scientists had evidence that the hormone called growth hormone might not be acting alone. The 1957 experiment opened an entirely new investigation. If growth hormone caused another substance to appear in the blood, what was that substance? And was sulfate incorporation the only thing it affected? Researchers began studying the circulating growth-promoting activity in much greater detail. Over the following years, evidence accumulated that these GH-dependent factors were associated with much more than the incorporation of sulfate into cartilage. They could stimulate processes involved in tissue growth, including DNA synthesis, protein synthesis, and the growth and development of cartilage. The name sulfation factor was becoming too narrow. It described the laboratory measurement that had helped reveal the substance, but not the much broader biological role researchers were uncovering. By 1972, a group of researchers, including William Daughaday, Kirsten Hall, Maurice Raben, William Salmon, and others working in the field, proposed a new terminology. They called these circulating growth-promoting factors somatomedins. The name reflected the idea that they were mediators of growth hormones' effects on body tissues. And with that concept, the original picture of growth hormone had fundamentally changed. Scientists had begun with a very simple model. Growth hormone enters the blood, reaches growing tissue, and stimulates growth. Now they had evidence for another possibility. Growth hormone could act first on the body. The body could produce another signal, and that second signal could then act on growing tissues. The pathway had changed from GH growth to GH somatomedin growth. But somatomedin was still a name for a biological activity and a developing family of factors. Scientists still had to determine exactly what these substances were. The search for the somatomedins continued through the 1960s and 1970s. Different laboratories were studying substances in blood that produced remarkably similar growth-promoting effects, but they weren't always calling them the same thing. Some researchers were studying sulfation factor. Others described activities they called somatomedin A and somatomedin C. At the same time, another line of research had identified substances whose chemical behavior and biological effects resembled those of insulin. They became known as insulin-like growth factors. As purification techniques improved, researchers began discovering that these apparently separate lines of investigation were converging. The molecule known as somatomedin C was identified with what became known as insulin-like growth factor 1, IGF-1. The name reflected an important clue about the molecule itself. IGF-1 shares structural similarities with insulin and can produce some insulin-like biological effects. But in the growth hormone story, its importance was much larger. Researchers now had a molecular identity for one of the major signals that had emerged from the original somatomedin investigation. And another part of the puzzle began to fall into place. The liver was found to be a major source of IGF-1 circulating in the bloodstream. Growth hormone could act on the liver. The liver could increase production of IGF-1, and IGF-1 could then travel through the circulation and influence growing tissues. More than 20 years after Salmon and Daughaday had detected an unknown activity in blood, scientists were finally beginning to understand what that second signal actually was. But once again, the answer would prove to be more complicated than the new model suggested. For a time, the emerging explanation seemed beautifully straightforward. The pituitary gland releases growth hormone. GH travels through the blood to the liver. The liver produces IGF-1, and circulating IGF-1 carries the growth signal to tissues throughout the body. This became the foundation of what was often described as the somatomedin hypothesis. But researchers kept investigating, and gradually, the simple liver-centered pathway began to expand. Scientists found that the liver wasn't the only tissue capable of producing IGF-1. Bone could produce it, skeletal muscle could produce it, and other tissues could produce IGF-1 locally as well. That introduced another important idea into the growth hormone story. Not every biological signal has to travel through the bloodstream from one distant organ to another. A tissue can produce a signal that acts on nearby cells. That's called paracrine signaling. Or a cell can produce a signal that acts back on the same cell. That's called autocrine signaling. So GH could stimulate IGF-1 production within tissues, allowing part of the growth response to be regulated locally. And the story became more complicated still. Researchers accumulated evidence that growth hormone also has important direct biological actions that do not simply depend on circulating IGF-1. This became particularly important for understanding GH's effects on metabolism, including its influence on fat mobilization, protein metabolism, and glucose regulation. The model was no longer one hormone makes the body grow. And it wasn't even just GH tells the liver to make IGF-1. Scientists were uncovering an interconnected endocrine system, in which GH could act directly, could stimulate circulating IGF-1, and could influence IGF-1 production inside individual tissues. The simple hormone discovered decades earlier had become a network. Look back at where this investigation began. Scientists had isolated a substance from the pituitary gland and called it growth hormone. The name suggested a remarkably simple biological story, but almost every step that followed made that story more interesting. Animal growth hormone couldn't simply replace the missing hormone in humans. Researchers had to obtain GH from human pituitary glands. Maurice Raben and other physicians then showed that human GH could restore growth in children with severe deficiency. That proved the hormone mattered, but it still didn't explain how it worked. Then Salmon and Daughaday took the investigation into cartilage. Growth hormone alone couldn't reproduce the response they expected in their isolated tissue assay. But serum from a GH-treated animal could. That unexpected result revealed another signal. First called sulfation factor, then incorporated into the concept of the somatomedins. And eventually, one of the central molecules in that story acquired the name we know today, IGF-1. Even that wasn't the end. Scientists discovered that IGF-1 could be produced not only by the liver, but locally within tissues. And GH itself could have direct biological effects. What began as a search for a single substance that caused growth had uncovered an entire system of communication between the pituitary, liver, bone, muscle, and other tissues. And that changes the answer to the question we asked at the beginning. How does growth hormone make an entire human body grow? Not by issuing one simple command. It participates in a network of signals that allows different tissues to respond, communicate, and adapt. But there was another chapter coming. Because for decades, the only human GH available for treatment had to be recovered from the pituitary glands of people who had died. Then, in the 1980s, that system would collide with a medical crisis. Just as a new technology was about to change growth hormone forever. Next Sunday, Part 4, what happened when growth hormone became available? Adaptation is survival.